Halo, selamat pagi semuanya. Apakah suara saya terdengar?
Dengar,
Dengar,
baik.
Mas.
Terima kasih. Good morning everyone. Bulan ini, bulan Agustus, kita bertemu kembali pada webinar bulanan yang diadakan oleh Pusat Riset Budidaya Laut Badan Riset dan Inovasi Nasional. Tema webinar pada Ocean Farm yang ke-10 hari ini adalah Sustainable Stream Health, Bridging the Technologies and Molecular Immunology. Today, I would like to uh, invite Rafael Castillo as one of our speaker. He's from National Cheng Kung University in Taiwan. And our second speaker is Mr. Wahyu Purbiantoro from Research Center for Marine Aquaculture, BRIN. But before we start our webinar, I would like to invite Dr. Supono to give a welcome remark before we start our webinar. Mr. Supono, please.
Halo, selamat pagi. Apakah suara saya terdengar?
Terdengar dengan jelas, Pak.
Alhamdulillah. Oke. Baik, terima kasih banyak Bapak Ibu. Selamat pagi.
Harus begitu.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi.
Salam sejahtera untuk kita semua. Om Swastiastu, Namo Buddhaya Kebajikan. Oke, terima kasih banyak Bapak Ibu yang hadir hari ini di acara rutin kita bulanan begitu, nggih, satu kali sebulan untuk berdiskusi, sharing terkait dengan sektor budidaya, terutama untuk budidaya laut. Good morning everyone. First of all, I would like to thank all of the participants today who has attending our monthly regular webinar discussing about specific topic in marine aquaculture sector. And of course, we would like to express our gratitude to our speakers today. We have Rafael Castillo from Taiwan, and he has been working mostly on the specifically on the disease management and mitigation, particularly in shrimp farming. And also the second one, we have Dr. Wahyu Purbiantoro, which is I believe that he is a colleague of Rafael Castello at some point, and he will also talk about the probiotic, paraprobiotic development as part of the mitigation and also controlling the disease in shrimp farming. And just a quick and brief reminder that this webinar has been conducted since the last year, and now we are at the 10th webinar, and hopefully, as also for the previous webinar, we expect that from today's session, we have a meaningful discussion, and then of course also impactful follow-up following the
Sepertinya jaringan Pak Kapus sedang lemot.
Ya,
Eh,
kedengaran.
ya, sambil menunggu Pak Kapus, mungkin we go to welcome remark from the head of Research Organization of Food and Agriculture, Ibu Puji Lestari. Kepada Ibu kami persilahkan.
Assalamualaikum warahmatullahi wabarakatuh. A very good morning, everyone.
Waalaikumsalam.
Excellency, the head of the
Good morning.
Center for Marine Aquaculture, Dr. Supono, distinguished speakers, Mr. Rafael Castillo from National Cheng Kung University, Taiwan, Dr. Wahyu Purbiantoro from the Research Center for Marine Aquaculture and the ruang, BRIN, the chair of this webinar, distinguished colleagues, ladies and gentlemen, and all the esteemed participants. It's my great pleasure and honor to welcome you to the 10th edition of the Ocean Farm Seminar Series organized by the Center for Marine Aquaculture. And today, we gather under the theme Sustainable Stream Health, especially bridging applied technologies and molecular immunology, a subject of critical importance for the future of aquaculture and global food security. First, so let's thanks to also Subhanahu wa Ta'ala for his blessing, and we still have a good heart to attend and gather in this important event at the Research Organization for Agriculture and Food, especially Research Center for Marine Aquaculture. Based on the theme or subject to be discussed in this webinar, as we know that Vietnamese shrimp is a key aquacultural commodity vital for fishery production, national economy, and export-oriented farming. Intensification and high stocking densities boost productivity, but also raise this risk, threatening growth, survival, and farmer income. Pathogens like bacteria, viruses, combined with poor water quality and stress factors make disease a major challenge for sustainable farming. Effective health management requires both preventive and curative approaches, supported by advances in agricultural technology, diagnostics, environmental management, biological agents, and immunostimulants. Integrating applied technologies with molecular immunology is crucial. So stream rely on innate immunity, and one promising tool is CpG oligonucleotides, which can stimulate immune response and enhance disease resistance. So as a part of effort to strengthen knowledge dissemination, experience exchange, and collaborative among researchers, academics, and aquaculture practitioners, the Ocean Farm tent serve as a platform for sharing innovation in stream health and farming. Under this theme, this highlight advances in disease management strategies from applied technologies to molecular immune mechanism, while promoting innovation that support healthy, productive, and sustainable shrimp aquaculture. So the Ocean Farm Sari Tents is very important and hopefully it will be very beneficial to share knowledge, disseminate research, improve understanding, and issues to be discussed today. Link between research and industry and foster collaboration among researchers, academics, and practitioners. With Bismillahirrahmanirrahim, officially I open this Ocean Farm edition. Have a good day and fruitful discussion. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Bu Puji, for the welcome remark. So before we continue to our speaker, I would like to go back to Mr. Supono as our head of research center. Please, Mr. Supono. Mr. Supono.
All right. So apologies to everyone that it seems that there has been a technical issue here, especially in my laptop. I'm not quite sure we are using the same internet connection. Mbak Debbie, Pak Idi next to me, they are fine, but unfortunately in my stuff, it's not working very well. But yes, maybe just briefly to sum up that Dr. Puji Lestari, as our head of the research organization for food and agriculture, has pretty much go through into the detail regarding our today's session. And I would like to add and also emphasizing about hopefully that following this webinar session that we can go through initiation of the research collaboration toward impactful outcome through the session. So that's all from me. On behalf of the Research Center for Marine Aquaculture, we would like to again express our gratitude and thank to our distinguished speaker today, Rafael Castillo, and also Pak Wahyu Purbiantoro, and also all Bapak Ibu peserta webinar hari ini. Terima kasih banyak. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Terima kasih Bapak Supono atas sambutannya. Thank you for Mr. Supono for the welcome remark. Before we go to the presentation from our first speaker, Mr. Rafael Castillo, I would like to introduce Rafael for our audience here. Rafael Castillo is from National Cheng University, Taiwan, from Department of Biotechnology and Bioindustry Science. Nice to see you, Rafael. I also graduated from NCKU from Environmental Engineering. And I think my professor also had some collaboration with your department. Nice to see you. And Rafael Castello actually has a lot experience regarding the white spot syndrome virus. He has a lot of experience about this disease, and today he would like to explain a lot to us as an audience about the development or the current situation from this study. So, Rafael, please take your time.
Okay, thank you for the invitation. Everyone can hear me. I think the audio works well, right?
Yeah.
Okay, awesome. Well, firstly, I would like to thank the BRIN, the National Research and Innovation Agency, for the invitation. Also for the Dr. Wahyu, also my colleagues, and I consider also my friends. I'm very happy to hear some good from him. So I'm gonna start sharing my presentation. Sorry. Okay. So I think everyone can see, right?
Yeah, it's clear.
All right. So nice. So I will start. So my name is Rafael Castillo. I'm also original from Nicaragua, so I'm from the Central America. I've gone from very far. But however, I also been interested about the agriculture, and then came here to aim some insights about it. So the topic that I'm presenting is about the strategies for disease control in shrimp aquaculture, more about from the research and to apply technology, most our lab is about. So my advisor is the distinguished Professor Han Jing Wan, and then this is the International Aquabiology Lab that is in Chenkong University in Tainan, Taiwan. So to start my presentation, firstly, I would like to introduce or remind this important concept that is the One Health. So this one is trying to summarize or point together like for the human health, there also need to be considered the animal, the plants, and the environment that we are living. So this is more an interdependent system that is one or this one might be weak or is being affected for some disease. At the end will affect us. So that's why it's important that for this one health, we also put attention in the or animal safe that can be by using biosecurity strategy, sustainable farming practices, or just by monitoring the disease. So that's one at the end will help us to we need to make sure about the environment, the animal we farm, the people all try to stay healthy together. One interesting about that one health fact is like in the world there are still remaining like 811 million hungry people that go to bed each night. It's been predicted that by the 2050 we're gonna be needing an 70% extra or animal proteins. And nowadays, more about our animal production is being that this loss is linking for specific for animal disease. Interesting also is about the global seafood production, more so starting from the 60 in less than 50 years, how being this one being increasing. And more beyond that, the difference about the agriculture and the capture fishery. So even more like a 52% in the 2018. This showing us that the agriculture is a highly promising solution for our sustainable future. However, the climate change effects is also threatening the agriculture. How? By the global warming, the sea level rise, the freshwater shortage also affect the ocean productivity. But one of these most important is that will be facilitate the outbreak for the currently disease that we know, and even for new pathogen or parasite that we still haven't faced or we do not understand them yet. In my lab, we're most focused about shrimp. So we know that shrimp farming is a major global industry. And interesting, more than 70% of this production is in Asian countries. This shrimp farming is aligned by the SDGs about the zero hunger, good health and well-being, climate change, and life below the water. So in our academic research, we're trying to get some insights on a practical strategy that can help us to reduce the shrimp farming loss caused by the extreme climate, to increase the shrimp production potential and the meat food demand of the growing population. However, there's still big threats that we are still facing that are the disease problem, also to find or to get a reliable stock and fry for shrimps. That's also very important. And I can say that we're still lacking a strong global like integration agriculture system, even beyond the frontier or different countries or like the public or private sector. This might need more integration for it. Here I introduce one of the manual of the diagnosis text for the aquatic animal. It is more presented for the WOA, that is the World Organization of Animal Health. So for this one, he described what are the main techniques for the correct detection of different disease that can be in crustacean, in here in a specific shrimps. So year by year, this should be updated from the oncoming diseases. But For all this process, there is table of experts that see, evaluate, and try to characterize how it really to detect this disease using to see the clinical science, PCR identification, or it can go to even for the qPCR, or the different kits that different company can generate. They also can need to be approved to follow these different guidelines. So here, for example, we present the APEN, the acute hepatopancreatic necrosis, even provide more guidance on what are the corrected tissue that we need to use for the detections. And there also for the Helicobacter pneumoniae, and the Taura syndrome virus, and the white spot syndrome virus that I'll be explaining more in this presentation. So this lab, the lab that I'm from, is a reference lab for white spot disease, and from the APEN, from the acute hepatopancreatic necrosis disease. So firstly, to identify or to get for all this process, we know that in the farm there is a disease. There is lethargy on the shrimps. There are like loss in the appetite, loss of movement, and we still seeing some clinical signs. So we know that something is happening. So after that, we need to go to try to identify and detect what is this main pathogen. If this is a virus, if this is a bacteria disease, or like try to understand this. And then when we go to the pathogenesis, is more to know like what's the molecular or cellular pathway that these pathogens are affecting in the shrimp. If this one is just hijacking or regulate all the metabolic pathway for viral replication, or these are secreting some toxins that are affecting or treating the shrimps, for example, with the apens. And then we go to develop some specific detections methods that can be used for or the PCR or the IQ or the histopathology, how is the main for or stomach, or can be pancreas or the intestine, how this really happens there. So as a white spot disease, this still remains one of the most deadly viral disease in cultural venomous shrimps. More the clinical science is start getting a red body coloration, and then we can see a white spot in the exoskeleton. They can be a high mortality in amount for 2 or 10 days after the infection, and infect not only shrimps but a broad range of crustaceans too. Interesting, the white spot syndrome virus, this is the main agent for this disease. This is an envelope double-stranded DNA virus from the white spot virus, and recently has been reclassified as a white spot virus shared by the ACTB in just these years. Here I am presenting a result for a cryogenic electron microscope that show us the empty packaging of the nucleosides and the completely packaging, and then we also see no tail about this intact period. Oh, in here, there's a QR code for this specific paper. So as we know, during this replication cycle, there's a lot of cellular and molecular things that is happening. Firstly, the virus had to enter the hot cells, most for the endocytosis process, and the where this one is inside, start for the DNA replication, also for the assembling, and then at the end for the virion release. This virus has more than 500 open reading frames. So 90% of this one has no homology for other viral proteins. So there have been understanding from some of the structural proteins. So what we're trying to focus in my lab is try to understand and classify this specific function of this novelty protein. During this pathway, there have been two interesting specific time points. One is about the 12 SPI. We can see here in this graph how the peak of the fold change really increased of the copies of the WSSB genome DNA, and the 24 for the late state of the infection. So we classified the 12 HPI as for the viral replication, and the 24 more related to morphogenesis, uh, morphogenesis pathway for in the virus. So in our lab, we're trying to focus describing different cellular metabolism that these WSB induced changes. We also try to standardize or propose more so in vitro methods to uh, characterize viral protein. We got, for example, the yeast-2 hybrid. For the yeast-2 hybrid, we try to target one specific viral protein, and then we have a library. This library might be different WSB open reading frames, or we can target some host proteins, or as a base, we can use the host protein. So when we have it in the plates, and then it's just only gonna be able to grow to a protein that activate the activation and the binding domain, and different promoter genes that help the yeast to grow in this selected media. So when we got them, we send them for the identification, and then we approve them with the selected plate that we can see here at the right. So trying to know that these two proteins show interaction. Then for this Western blot image, I show it here, that we're using the SS9 cells. This is most about the insect cells. This is that has for this similarity with the shrimp line. And then we try to express uh, viral protein and also host proteins. And then we use immunoprecipitate using some also, in this case, flat pits, and this one is able to pull down the other proteins. Is a test that show us interaction. Then another things that we're doing is using a affinity purification, the mass spectrometry. So what we explained in before, we express our recombinant protein, while at the same time we've got some WSB infected gills, and then we got this protein lysates, and then we're going to combine them, and then after some washings, we're gonna have our protein with the specific host protein that we would like to identify. So we send this one to the LCMS, and then we got some bioinformatic analysis, and also there's some data filtering, and then after that we go for the KGG pathway analysis. So we do this one in order to know direct and indirect interaction with this specific viral protein. And even we're able to find some other, because of in low abundance, but we get to know other viral protein. For example, in this case, for this publication, they're being targeted to study the 004 that had been found to interact with the lactose dehydrogenase. So uh, uh, we are able to prove that different metabolics are being hijacking for the WSSB. That can be the glycolysis, the glutamate, the nucleotide biosynthesis, and then we try to go more deep to find the corresponding proteins. We mostly hypothesize that this metabolic thing is happening by protein to protein interaction. Now, because we had different database, we got transcriptomics, we also had proteomics database. So in our program that we got, that is the ComtiView, that we are able to form differential expressions of more than 7,000 genes between infected and non-infected at 24 hours. So for this correlation, we're gonna have like the red that is for the positive correlation and the green color for negative correlation. And after that, we try to map the in this case, the 004 that we got from the AP mass for the affinity purification list. And then we create we got the network. For example, in this case, we see this big, like a red network. We see some specific hot genes that are there, and the color we see two different kind of red. These one are related to ribosome biogenesis and for the nucleocytoplasmic transport. When it gets a little more interesting and complex, and then when we have negative and the positive correlation in the same network. So trying to tell us that this hub with interaction with others during this infection is being upregulated and downregulated. So however, this one is it's a correlation network that later on when we find this hub gene, it has to be um, reconfirmed that uh, that the role and the function is also being triggered by the WSSP. However, this platform really helps to in a short time we can target some specific pathway or to understand, for example, for the nucleocytoplasmic transport, and then try to see how this protein is linking different proteins can be expressed in just 2 hours, 6, 12, and uh, can contribute maybe for a more fast diagnostic or to check or evaluate some host or enzyme activity. So the idea for all this research is, well, previously there had been, this is a very huge project. It's a multi-omics established. It's more by our team, and this had like a long time that had been working and preparing. So firstly, is try to get the shrinks with the WSSB genomics. So in this case, we're being more targeted about the Pinemus monodome, that is the black tiger shrimp, to have the sequencing the whole genome for these shrimps. Then we also have some transcriptomic database from hemocytes. Also, we got the from stomates too, from different time point 1, 6, 12, and 24, so we can compare it and see, depending by the time point, how the changes are happening. The same by the proteomics. Then we're trying to work with the interactomics. Then when we have some protein interaction, that we know that this protein is not working alone. So it's gonna be forming a multi-complex that can be linked from viral to viral interaction or viral to different hosts that, like we're showing by the AP mass for that direct or indirect interaction. And then we would like to link this multiplex complex with to the specific metabolites. We also have some metabolomics work in different time points. And all of these one, at the end, we're gonna help us to describe or characterize more better with the shrinks with the WSSB, the phenotypes. After, for example, we get some genes for the correlation networks, we can do some silencing, we can do some gene overexpression in pathogen challenge to see how the WSSB phenotype change. If the virus is decreased or this can uh, enhance the replication of what is really happening there. So this has been working for more at an active international like cooperation. We got here my Professor Hans Wang. These are the professor from Taiwan, professor from NTU, Siswan Lin, Hao Qing Wan's too, with different expertise for this, for the Ian Lee from the metabolomics and the synthetic biology. That also we had some other professor from overseas, like the Dr. Maria Bet from the Philippines, the Professor Subha Basu from Malaysia, and professor from the Thailand, also expertise in proteomics and the interactomics, Aku, Professor Senapi, Professor Laya, and the Professor Aran also from the United States, and the, the distinguished Professor Dr. Iko Hirono from Japan. So to get in detail about the interactomics, so we would like to study all these 432 different open reading frames, and then get to know the virus and virus WSSB protein complex. Then for the M2 is mostly to see this complex both with host protein and viral proteins too. And then the other is try to target the complex with a specific metabolite. So this one at the end will help us to construct more a comprehensive network, and at the end will help us to validate all these the accurateness from these hub genes or the hub proteins, and investigate later on their biological functions of this one. At the end, this one has a really scientific impact. Must to be the first invertebrate like double-strand DNA for have like a interatomic network, and to break down it connects technology and wants to pursue the world's leading shrimp and pathogen research in Taiwan. For also have a social impact to select preferred commercial traits that can be identified from this work for shrimp breeding, for reduce disease outbreak, and also for increase the profits and adaptation for the climate change, and to reduce the economical losses. At the end, the main point of this, when we studied trends, patterns, and the environment into playing for possible disease-resistant shrimps. So when we identify these resistant biomarkers, that we find them in a WCB resistant shrimp for natural population, now we had a specific pathogen free from the nuclear breeding center that I'm gonna introduce in a bit, that also might be later on resistant shrimp and contribute for the sustainable shrimp agriculture. Sorry. Now, for the next part, I would like to introduce some detection kit or some technology that had been releasing for the contribution from some companies here. One of this specific company, I think most of might be familiar, is the GeneRich Biotechnology Corporation. So this is a PCR diagnostic system just for event causing plasmid. So this target or using, sorry, 3 primer sets that for the one sequence for the plasmid as a name as A1, A2, and the specific the peer for the BRP region of the PBI1. So this is just a kit for a PCR. We already have all the reagent and the primer there, and also provides some positive with countable copies that's gonna be like for the 2000 or 200, so like a positive control, and then we also prepare the negative, and the different result depending on the band. For example, lane 4 shows like which is toxin number 1, and then 5 lane 5 that's a sample with no viral apen plasma with the toxin deletion. Another one of this one is like a pod kit test for rapid and easy detection of the white spot syndrome. This insulate isothermal PCR. So it's mostly like at these machines. It has all we got the tissue. We had to extract the DNA and then run this one. I think less than an hour, it give you the result. It's mostly for the weight line that the machine had. And then this one that I find it more interesting is to generate WSSB RP rapid test kit. This one was with the Ino created company. So the idea is Here they are selecting some gill tissue, and then they put it like this COVID test. But before doing this one, there were start to study the specific non-structural protein that they want to target, to develop the prototype, and there had been a different tests that had to be done, and has to be aligned with all the OIE requirements for this test. So this as the first LSIA rapid test for this diagnostic kit for aquatic animal, even also approved for the world. So after just we got the tissue, there are different reagents. We put this one. Most of us are familiar, and then we got the two bands that offered positive, and we just only got once is just like at the control for the test. We're just waiting for dispense 5 to 6 drops and between 15 and 30 minutes to check the result. So in here, lastly, I would like to introduce or share with you guys about the center. There's an international center for the scientific development of shrimp aquaculture and how it is that these breeding programs. So this center is since the 2016 and is being the first integrated shrimp breeding center in Taiwan and one of the world leading as a specific pathogen free for tiger shrimp and as a genetic resources. This is also a non-profit center, and with the strong integration and politics is mostly research and has also cooperation with industry too. So the idea for these centers is to have some breeding strategy that at the end can enhance the productivity, resilience, and the nutritional value. Also to strengthen uh, the preparedness of the resilience to disease, climate change, and also supply disruption, and to ensure the global food security. This monodon shrimp is classical economical important shrimp species in Asia. It also was suited for aquaculture with a high economics value. So in these centers, this center is in two different area. One is in Thailand, and the other part is in Hualien, more for like a big production. However, with the shrimp nuclear breeding, there is for the broodstock and for the larval production, where is controlling natural mating, broods to condition or maturation. We're being trying to apply some AI camera to help us to know the correct size, how much they are eating, how much they are growing, and even for the ovary maturation. And the more bigger center as a multiplication center is like try to um, thinking this as a commercial way, but still as with academic purpose. It's like uh, we had the we developed the broodstock. We also got the larva production, but how this one is responding, how this one is acting as a in a commercial plan. It's we can develop like a protocol or the techniques for farmers, how being the correct conditions of maintenance for this. So it giving us some inputs to get the disease surveillance and even collect some of this one for some diagnostic and make sure that even in a commercial level they can still be as a specific pathogen free. Another interesting is we also are trying to focus about the nutritional part or using some supplemental feed. In this case, we're trying to study the perilla frutensis, how it can enhance the immunity, microbiota modulation, or tissue protection during AIDS, and also for this new common disease, the TPD, the transfusion post-larval disease, and for also WSSB2. And this also, we got intervention and outcomes about the perilla, that is the plant derived bioactive, also some use for the nanobody, that is the engineering of probiotics for virus neutralization, inhibiting their replication and microbiota balance. Another part is applying with the AI system is for the smart management platform. That is for the ovarian maturity assessment to try to help or to see or to identify the ovarian maturation. And to I was saying about the see the shrink biomass, the size, dot fullness, how much this one is eating to try to reduce the low carbon waste during the feeding management. So more beyond that, for the nuclear breeding center, we already had some family established and well started. There are families for different purpose. For example, for low salinity, high salinity, uh, high growth, low growth, or like high a different degree of temperature too. And had been tested by 5 or 7 pathogen detection. This is just as an example if we would like to enter or how this family were established or accepted. You know, there has to be firstly a primary quarantine that when we screen for all these pathogens. If they had contained any of these ones, so it has to be to discard. We cannot use that. If there are if they are passed, then they go to the secondary quarantine facility that are still for the observation, and then we go to the first one generation. And for this generation, they had been tested again for this pathogen, and then they can be a part of us as a one of the family, and then we keep their surveillance in the center. Interesting is like we had been starting to have like a collaboration. This is or like to create this center but in other countries. This is an example in Brunei as a satellite center, as an academia industry collaboration. So what mostly we're doing in these centers, we're trying to replicate and doing the same in the Brunei facility. Here, there's also the pictures from the for the multiplication center, that the big center for a big production, where that's on a standard for the harvest. And we're trying also to develop some for the post-harvest part, right? Like after we harvest them, how it has to be the correct way to prepare them, and even also from to see the taste, you know, for the last part that's not really about the production, but more about the quality of the products. So thank you very much. I mostly finished my presentation. So here we're trying to keep this eco-friendly, sustainable shrimp agriculture. We believe that this shrimp can be one of the targets called animals, livestock, like to as a comprehensive strategy that might involve a collaborative effort into the global shrimp research community. So I appreciate but this is me with my lab mates and my professors, and of course the professor Han Wan. And thanks a lot for your attention, and I think I can take in any questions.
Thank you, Rafael, for the good presentation. It's such a good information for us, especially for the development and the current situation of this white spot syndrome in the stream, and how you can integrate the AR, and also how you make the detection to detect this virus. Before we look into the chat section, any question from the audience? Maybe you can raise your hand from the Zoom. Any question, or maybe we look into the chat section. We have a question for Dr. Castillo. The first question is from Professor Rahman Shah. His question is about, the first question is, moving from reactive to preventive disease management, how can the shrimp industry shift from responding to outbreaks toward risk-based prevention involving the SPRC, biosecurity, surveillance, water quality management, microbiome management, and enhancement of host health? And what's the gap? And why some of the disease control technology perform well under laboratory study, but sometimes it's inconsistent in the commercial farm? Maybe you have comment about this, Rafael?
Okay. Let's try to share or discuss some insight about it. So the thing is, mm, maybe we can try to make a difference, right? We got the SPF, that is a specific pathogen free, right? And then we might have as R or SPR, sorry, that is like a specific resistance pathogen, right? So there are two different lines. So the resistant line is might be one that, for example, that can be our goal after what identifying some biomarker for all the molecular study, and then we see the phenotypes, how the virus are decreasing, or in case some different metabolite or some immunity, that are immune resistant. But what happens with the free pathogen is this is a free pathogen. So we got a free pathogens, and then we just put it in a management or in a tank.
Dengan tas ini,
that
bawa makanan.
sorry. Okay,
Okay,
so
you
it
can continue.
yeah
Sorry.
yeah it would put in a tank, or maybe with the density or with the oxygen or other parameters, or when there's a focus of disease there, uh, it's very possible that at the end they might be infected. So that's why keeping the environmental condition, I can say, is really crucial to understand how is the correct um, density of animal that we can use in the farm. This is hard, of course. This is a challenge, and the outbreak for this one is sometimes, or in a specific WSB, is related to the increases of the temperature. That's why in some countries, in some area, we can see more WSB in also some specific time, but in other places we don't see. And what normally farmers do, and they see an increase affection of WSB, is mostly they just harbor this, right? Even you have the different things. But then after that, then we know we might have to put this one in quarantine, remove all the water, use the sterilization correct process. And then after that, when we put more of this one, we need to keep check their healthy condition. But I think that's the thing. But how many centers can we have that they can provide us a free pathogen free? That's kind of one of the big challenge for this, you know.
Okay. And maybe next question is, so actually, which factors are the most responsible? I mean, how we can apply the method, the environmental condition, the pathogen pressure, stream light stage, or farming system?
Okay. But I think the monitoring, the surveillance is really important. The thing is, like when we really have a big tent of area, it can become difficult. But uh, mostly in my experience, when I got into a tank, and there I saw that there are some shrimps that not really eating. I just try and check the waste, and there's like an accumulator of the feed. I can see that there are lethargies. They're not really in movement. They just kind of stay by. Even if we take them and we don't see any uh, um, pathogen signal, right? So it get on to know that something is happening there, right? So they also sound like a small wet lab technique that we can see to see how the organs just with a normal a microscope, but it get off the attention that something is happening. How? So then how might be like to prevent this? Of course, the supplementation feed help us. There now in the market there is a lot of probiotics, also functional feed that in a way uh, can modulate that innate immunity, like make the shrimp a little more resilient into one disease. But it also is not saying that it's like a drug or therapy that it's going to save us. We see the disease in the tank, and then we start using the this probiotic or something, and no, it's not going to save.
Okay, thank you for the answer. We go to the next question from Mr. Suryadi Saputra. Question for Mr. Rafael. I'm currently conducting research on the freshwater adapted Vanami shrimp, and I have not yet studied the various diseases you mentioned in your presentation. My question is, can those diseases still affect freshwater adapted shrimp, or do you have any experience with this kind of research?
Uh, sorry. Can you repeat it?
He's asking because right now he's working on the freshwater adapted Vaname, and he has not studied about the disease you just mentioned in your presentation. So his
Mm.
question is, can those diseases still affect the freshwater adapted shrimp? Do you have any experience with this kind of research?
Well, because I normally we work with the shrimp with 20% of salinity, right? So it's more like a bright fixed water. So to say the fresh water, I cannot give an straight answer, but what I can say is in the future, some another disease um, might be an outbreak of that, right? Also about the raising of the temperatures can be effects on some of this one disease, and we might need to put attention on that.
Okay, I hope that can answer the Mr. Supreme question. Maybe we go to the next question from Miss Mulyani. Question for Dr. Castillo. Currently, which one remains the primary cause of shrimp disease globally? White spot syndrome or AHPND? And are there any existing technologies capable of preventing this disease at lab or the field scale?
I can say yes that these two are still one of the main threat. However, now there's like a more different or oncoming thread. For example, I just introduced the DPD, the translucent larvae, but this one is more for the fry, for a larvae disease, for really a small of shrimps. Yeah. And the EPEN to be also a bacterial disease has more threat for kind of to 1 gram to 3 grams, more like a bigger shrimp tends to have a little more resistance. However, with the WSSB, mostly target more kind of more bigger shrink are like a more susceptible. The 3 grams, 10 grams gonna be very lethal for them. And the best is to keep the monitoring. Also, one of the problem that I'd explained is the lacking of integrating system for the aquaculture. I mean, if we got a farmer that has an outbreak of one of disease, and then if he in a way can report, or let everyone know, then we in a way can track where was the stock fried, and then we can try to link other farmers that are using the same, and they can be in quarantine or like a being alert. But is this kind of communication is missing, is making hard, because then the for where they got the stock or the fry, people will buy them again. So that get more about the how we really do this business, right?
Okay. Maybe last question before we go to Dr. Wahyu presentation. One question from B22S. Question for Dr. Castillo. What are the main differences between AHPND and TPD, especially regarding their virulence factors and mechanisms?
HPND. Where we got these questions?
The TPD and HPND. Is there
Oh,
any
okay.
differences?
Got it. Okay.
Yep.
Well, this is one of the focus that we're trying to investigate, because um, we find that uh, these two diseases are also from the bacteria and for the some different strain of people. So we know for the apenet, there is the existence of a plasmid that is containing the two genes for the peer A and peer B toxin, and they're gonna be together to form the toxin for the disease. So this TPD, that also has a plasmid. We're also be working for the whole genome sequence to understand this TPD. I think we got a publication that is mostly at the end of this part for the final revision and to be published, right, to free. And there are also some toxins there, but they are completely different by the ape. And we're trying to see which are the specific vibrio species that can contain it or can produce it. That's at the end is the challenge for this bacterial strains. Uh, mm, they might be kind of similar with others, but they at the end won't produce the toxin, or they contain the plasmid, but they do not spread the toxin. So there is something happening there that also need to be well understood. So in order that we diagnostic the disease, we know that this is the shrimp is infected with this bacteria that contain this plasmid and will express the toxin. So that's kind of the challenge part, because if we just only identify the bacteria, but we see in the field that there is no toxin, there is no disease. So that's why all these WOA standard keep trying to be very demanding for a correct diagnosis in the disease.
Okay, thank you, Rafael, for the answer and also for your presentation. Maybe we still have some question in the chat box here, but I think we should continue to our next speaker. Maybe Rafael can answer from the chat box if it's okay for you.
Yeah, I'll
Uh,
take a look. Yeah. Okay.
okay, thank you so much. So thank you for Rafael for the good presentation. And we go to our second speaker is Mr. Wahyu, Dr. Wahyu is the staff in our Research Center of Marine Aquaculture BRIN, and he's worked more about the development of probiotic and parabiotic, and he also worked a lot in the aquaculture and the aquatic animal health. And today he will present about the effectiveness of dietary heat killed by uh, bacillus, something related to the CPG audience. Actually, I'm not really familiar with this term, so please welcome Dr. Wahyu to present his presentation. Yes, Dr. Wahyu, maybe you can start your presentation.
Okay, you can see my presentation, Nova.
Yeah, yes, too small, but maybe you can make it bigger.
Right. Okay, now.
Okay, you can
Okay.
start.
Okay, good morning, everyone. Before I begin, I would like to extend my sincere respect to the head of the research organization for agriculture and food at BRIN, Ibu Puji Lestari, and to the head of the Research Center for Marine Aquaculture, Bapak Supono. And I also want to give a special greeting to my good friend and fellow speaker today, Rafael. It's so great to share this virtual state with you again after our PhD days in Taiwan. And of course, very warm welcome to all the participants joining us today. Thank you for your time, and I'm very excited to share this research with you all. Okay. I want to introduce my name. My name is Wahyu Biantoro, and I'm a researcher at the Research Center for Marine Aquaculture BRIN. And today I will be presenting our research on the potential of CPG ODN in disease control for Vietnamese shrimp culture. And we are exploring an innovative approach to boost the shrimp natural defense against devastating disease. Okay, next. As we know, let's start with by looking at the big picture. We know shrimp aquaculture is a massive global industry and currently value at over around 81.5 billion. And the white leg shrimp is the true backbone of this industry, accounting for nearly 52% of all global crustacean aquaculture growth. Right. However, what it is, this rapid growth face severe bottleneck and outbreak, particularly from pathogen like the white spot syndrome virus and bacteria causing acute hepatopancreatic necrosis disease, lead to the massive economic losses every year. And finding effective ways to control this disease is the most critical challenging facing the industry today. And before Rafael already explained how to what it is work with the VSSV, and today I want to share with you about our lab during my PhD study in Taiwan. We what it is develop some tools to boost immunity on the shrimp. And we know to understand how to fight this disease, we must understand the crustacean immune system. And as you know, unlike vertebrate, which have both innate and adaptive immunity, crustacean fundamentally lack an adaptive immune system. This means they cannot develop traditional long-term antibody memory. And if a shrimp survives an infection, it doesn't build up specific antibodies to fight it off faster next time. And because they rarely on their innate immune response, our goal is to find a way to prime stimulate this system, and this is where CpG-ODN come in. This CpG-ODN can act as a potent immunostimulant and the artificially trigger toll-like receptor in the shrimp, essentially putting the innate defense system on high alert without needing adaptive memory. And so what exactly are CpG-ODN? CpG-ODN is a short synthetic single strain DNA molecule containing unmethylated CpG motif. This specific pattern acts as a pathogen associated molecular pattern or PAMs. In nature, these unmethylated patterns are common in microbial DNA, but rare in the vertebrate DNA. Therefore, when the shrimp immune system detects them, it recognizes them as a bacterial threat and launch a defense response. And you can see what it is in the figure in here. And this is CpG, unmethylated CpG dinucleotide. And however, CpG-ODN in the native form with a standard post-polyester have a limited. They are rapidly degraded by nucleus inside the body, host body. To make them effective as a treatment, need to modify them, and by changing the native PO backbone to post-proteated or PS backbone. Essentially, just adding a sulfur and to create a nucleus resistant molecule that significantly improve its stability in vivo. Okay, next. And how does the CpG-ODN work at the cellular level? This diagram illustrate the proposed mechanism. First, the CpG-ODN are taken up in the cell, like through clathrin-mediated endocytosis or macropinocytosis. Once inside endolysosome, they are detected. Interestingly, incrustation high mobility group protein act as the first sensor binding the CpG-ODN, and this then trigger a unique signaling pathway activating a toll receptor on the membrane and ultimately leading to the activation of immune defense genes. And then, okay, we know the promise of CpG-ODN here is significant, and some studies have shown that synthetic CpG-ODN are highly effective immunostimulant in crustacean, whether administered via injection or orally. They significantly enhance immune response, including phagocytosis, hemocyte production, reactive oxygen species generation, lysozyme activity, and the propanol oxidase system. However, here is the paradox. While highly effective in the lab, synthetic CpG-ODN are simple to expensive for commercial shrimp farming. The cost of synthesizing and purifying them to remove endotoxin make them financially unviable. And during during my PhD study, our lab innovation addressed this directly with what we call HKBS P60 innovation. It consists of three parts. One part is the pilot shuttle vector plasmid engineered to carry exactly a 60 copy of active CpG-ODN 166H motif, and the carrier, we use Bacillus subtilis, a safe gram-positive bacterium to massively propagate the plasmid. And this biological production eliminate the need for expensive synthetic purification. And the third, the safety lock, comply with GMO regulation and ensure safety. We heat kill the bacteria by boiling it, and the bacteria are biologically inactivated, but the structure integrity of the cell wall and the plasmid inside is maintained, allowing it to safely deliver the pilot. Okay, next, to test the innovation, actually we designed a two-phase translational study design. The phase 1 molecular proof, we use in here we use reverse scaffolds to deliver the P60 plasmid directly into the anterior midgut of the shrimp. And this allowed us to study the raw transcriptomic and tissue-specific immune responses without interference from feed. And the phase 2 for practical proof, we stimulate the we stimulate farm condition. We coated a commercial feed with the heat kill Bacillus subtilis containing P60. Containing P60 followed by a lethal Vibrio Harvey challenge. Next, in here you can see the reverse Gavard technique used in the phase 1 by delivering the PPS solution containing a microgram of the P60 plasmid directly via the anus. We bypass the stomach and ensure it reaches the midgut, avoiding interaction with a feed. We then sample the hemolymph, hepatopancreas, and midgut collected at 6 and 12 hours post reverse Gavard for RNA sequencing and real-time qPCR validation. This slide detail the construction of our pilot. We started with the PHD4325 vector, and also we call this P0 is empty plasmid, and this we use as a control, and inserted the sequence containing 60 copies of the CpG 1668 motif to create PSD 4325 60 CpG, our P60, and then we get the electroporation confirm the correct pen size, finding the successful integration in the 60 CpG copy. In here, we can see in the pen this pen size of 60 copy of CpG, and this pen of size of PAD 4325. This is the vector. Okay, the results in here. We moving to the results of phase 1. The RNA spacing data from the midgut 60-hour post stimulation reveal significant transcriptomic awakening. The heat map and fold chain data show that compared to the control, P60 treatment significantly upregulated numerous gain across various immune functional categories, particularly those related to the microbial recognition, the propose system, and the antimicrobial peptide or AMP genes. And we also validate this finding with the real-time qPCR, focusing on antimicrobial peptide. The result are streaking. We observe a significant spike in the expression of IMPS, specifically PAN3 and ILF, particularly in the hepatopancreas and midgut at 6 hours post administration. And this confirm that the P60 successfully penetrate midgut cell and activate a broad spectrum innate immune response at the genetic level. And in the phase 2, we try simulating farm scale application. We move to 28 a day dietary regimen, and shrimp were divided into the 3 groups: a control feed coated with the heat kill Bacillus subtilis contain the MT factor, and feed coated with the heat kill Bacillus subtilis contain the P60. And we have a note in here: Bacillus subtilis containing plasmid is completely inactivated by boiling at the 100 degrees Celsius for 30 minutes. And after 26 days of feeding, they were a challenge with the lethal dose of Vibrio harvey. We then track mortality and biomarker over the next 7 days. And this one, before the feeding trial, we confirm the successful transformation and heat inactivation. And this fluorescent microscopy image show Bacillus subtilis expressing GFP. Image C confirm that our Bacillus subtilis P60 strain successfully carries and express the plasmid prior the heat inactivation. And then the phase 2 result about the growth and survival baseline. As I mentioned, this table confirm that the dietary supplementation has no negative impact on growth, final body weight gain, specific growth rate, and survival during the 28-day feeding phase were scientifically similar across all treatment. And this proving the baseline safety of the heat kill Bacillus subtilis contain P60 feed. And then we have the result about we also monitor the key immune system, the key immune enzyme in the plasma during the pre-infection priming phase, day 40 and 28 of feeding. We saw significant spike in alkaline phosphatase and acid phosphatase activity in the heat kill Bacillus subtilis containing P60 plasmid group. And furthermore, post-infection at 12 and 24 hours after injection with the Vibrio harvey, the heat kill Bacillus subtilis P60 shrimp exhibit massive accelerated surge in acid phosphatase and alkaline phosphatase activity compared to the control, indicating a prime and ready immune system. Then, and the most crucial result is a survival after the challenge. Input the high dose and the low dose challenge. The heat kill Bacillus subtilis containing P60 group so in significant lower cumulative mortality compared to the control group. For example, in the high dose challenge, mortality was reduced to 60% compared to 90% in the control and 100% in the MT factor group. This clearly demonstrated that dietary supplementation with the heat kill Bacillus subtilis contain P60 translate to a significant survival advantage against the active Vibrio outbreaks. And then to synthesize our finding here, the proposed mechanism of action for heat kill Bacillus subtilis containing P60 plasmid. In the step 1, protected delivery, the heat kill B subtilis act as a shield protecting the plasmid through the digestive tract. And the step 2, intestinal uptake. In here, the plasmid enter the midgut cell where the CpG motif bind to the TLR. Then step 3, transcriptomic awakening. That trigger the upregulation of key immune genes via nucleus factor kappa B and or protein kinasasi pathways. And The last one, systemic axial. In here, upon detecting a pathogen like Vibrio, this PRIM system analyzes rapid amplified release of detoxifying enzyme and antimicrobial peptide, leading to enhanced level of alpha-NAMI. And this one, future research roadmap. Actually, after the research, we have a plan to do comparative motif analysis to understand sequence preference across different crustacean species, and then we want to what it is, a deeper transcriptomic mapping to build proteomic expression profile, and then we want to do a long-term memory trials to investigate the potential for train innate immunity and establish the duration of protection. And the last one is in, of course, a field scale validation. We want to deploy this bioengineer vector in the commercial setting and evaluate the real-world impact on cost, safety, and yield. Maybe. Thank you for your attention.
Thank you, Dr. Wahyu, for the presentation. Before we look into the chat box, any audience want to raise a question directly to Dr. Wahyu? Please, we are open question session for now. While waiting for the question, we have some question from Professor Rahman Shah to Dr. Wahyu. The first question is apakah peningkatan ekspresi gen imun setelah pemberian CpG-ODN benar-benar berkolerasi dengan penurunan patogen load dan juga peningkatan survival, bukan hanya menunjukkan adanya respon molekuler? Apakah ada evidence-nya nih Pak? Bagaimana? Pertanyaan pertama.
I answer in Indonesia ya.
Boleh.
Ya, dalam studi ini ya kita tidak mengukur penurunan patogen load ya sebenarnya. Tapi untuk peningkatan survival ya kita melihat peningkatan survival dan memang ada peningkatan survival di sana. Seperti itu Mas.
Oh. Berarti yang dilihat dari peningkatan survivalnya Pak ya, bukan dari load
bukan
dari
dari patogen load. We only what it is see the increase of survival rate, and also we measure the response molecular after the treatment and after the infection like this.
Lalu pertanyaan kedua dari Prof. Ramansyah untuk Pak Wahyu. Menurut saya, salah satu isu terpenting untuk aplikasi budidaya, bagaimana sebenarnya CpG ini dapat diberikan secara praktis. Jika injeksi, mungkin efektif untuk tahap eksperimental saja, tapi bagaimana dengan oral delivery yang dapat melalui pakan, enkapsulasi, atau mungkin ada inkorporasi dengan nanopartikel, atau metode lainnya yang bisa diaplikasikan di tambak?
Ya, untuk ditambak sih praktisnya ya kita menginkorporasikan dengan pakan. Jadi untuk studi ini injeksi itu kan hanya untuk molecular proof like this. Bagaimana ketika kita hanya memasukkan plasmid containing CpG ke dalam usus udang itu tadi, apa yang terjadi itu loh. Jadi big goals-nya seperti itu. Setelah kita mengetahui apa yang yang di regulated atau apa yang di unregulated, baru di sana kita mentransformasikan si plasmid itu ke dalam bakteri. Dan bakterinya ini kita gunakan untuk mereplikasi plasmid, dan karena kita menggunakan bakteri gram positif di sini, jadi dia kan cell wall-nya tidak mengandung endotoksin, sehingga we need to no need to purification, dan kita bisa langsung mengaktifasi si bakterinya, kemudian di-coating ke pakan dan diberikan ke tambak. But sebelumnya we must make sure the bactery shape dan the bactery form itu tidak lysis. Jadi kita cek dulu, bakterinya itu tidak lysis, tetap utuh. Jadi kita bisa memastikan bahwa plasmidnya itu masih ada di dalam bakteri itu. Baru kemudian kita coatingkan ke pakan dan pakan itu diberikan ke udang. Seperti itu, Mas.
Nanti di sini host bakterinya itu sangat penting juga, Pak ya, sebelum kita aplikasikan si PJODN tadi.
Iya, host
Lalu
bakterinya
di
penting
sini,
di sini. Iya, host
baik.
bakterinya sangat
Lalu,
penting di sini karena pada umumnya kan ketika kita memproduksi si PJODN itu kan kita menggunakan E. coli ya, metode konvensionalnya. Jadi kalau kita menggunakan E. coli untuk purifikasinya itu akan mahal karena kan kita harus menghilangkan endotoksin di sana. Kalau untuk keperluan pengobatan untuk di manusia is oke, tapi kalau untuk di aquacultur itu kan ya terlalu costly gitu kan. Jadi bagaimana solusinya ya? Kita menggunakan shuttle vector plasmid yang mana dia bisa replicate di E. coli dan juga bisa direplicate di bakteri gram positif. Di sini kita menggunakan Bacillus subtilis yang terbukti aman dan safe gitu kan. Jadi ketika dia udah
baik.
direplikasikan di sana, baru kita panen dan diberikan ke itu tadi ke udang dengan di coatingkan ke pakan, diinkorporasikan dengan pakan. Seperti itu, Mas.
Terima kasih atas penjelasannya, Pak Wahyu. Lalu pertanyaan selanjutnya, apakah si PJODN ini lebih efektif diberikan pada fase apa? Di broodstock, Novillus atau post larva diaceri, tahap nursery, atau selama proses pertumbuhannya?
Saya kira tergantung dari itunya tujuan kita tadi. Kan intinya kan tujuan kita itu kan adalah untuk menstimulat innate imun sistem yang ada di udang. Jadi kita harus mengetahui dulu kapan sih udang itu rawan terkena penyakit. Jika dia rawan terkena penyakitnya saat dalam bentuk PL ya, pas waktu PL itu mungkin 30 hari pertama atau 60 hari pertama ya kita aplikasikan itu tadi CPGODN. Tetapi beberapa penelitian yang telah dilakukan dan ini ada beberapa penelitian yang kita tidak publikasikan itu ketika CPGODN ini diberikan secara terus-menerus malah dia juga mensupres menekan immunity. Jadi perlu dilakukan itu interval pemberian CPGODN ke udangnya juga untuk memberikan efek yang lebih optimal. Mungkin seperti itu.
Baik, Pak Wahyu, terima kasih atas jawabannya. Dari audiens, ada yang ingin bertanya secara langsung? Kami masih membuka sesi tanya jawab. Jadi nanti bisa bertanya langsung baik kepada Pak Wahyu atau juga kepada Mr. Castillo. You can ask the question directly from our Zoom. We still open for the question section. We have another question from Professor Rahman Shah. This question is for Dr. Castillo and Dr. Wahyu. Maybe both person can have different opinion about his question. And the question is, if we want to build a sustainable stream health management system for the next 5 to 10 years, what should be the priority? Better pathogen control, stronger host immunity, better environmental management, or integrated combination of all three? And what research should be start today to achieve those three? Maybe Mr. Wahyu or Mr. Castillo can answer. Both can reply. Feel free to answer.
Oh, maybe I can go first.
Please.
Okay, thank you. Well, I can say yes, that will be for sure our main objective. Like try to have a shrimp line that have a good pathogen resistance, has a strong immunity, and can be adapted to different environments. Even I think I read a question before that was asking, can we have a shrimp line that has fast growth and has like a high pathogen resistance? So the thing is, uh, that is what we expect, right? But at the end, at the breeding program, it's not so easy. It's not so easy to achieve it, because then firstly we had to describe or identify the genotypes, and then try to target what are the genes in this pathway that they are involved, for example, for the fast growing. And if these one are linking to the shrimp resistant, maybe they can be part of a breeding program that they can be selecting both traits. But if they are kind of in a opposite way, then at the end we might have to make a decision that what might be more important at this time, to be maybe an APEN resistance, or like a WSSB resistance. For the immunity system, we can try to use some like herbal supplementation, or some of the heat shock CPG, like what Hugh is presenting. And from the environmental management, I will be also say um, sustainable farming practices, or like a biosecurity also, to like we make sure that we have using a healthy fry, and then in a way we monitoring our shrimps conditions, um also check that the oxygen, the nitrogen level. There's also some probiotics that help us for the water quality. And yeah, at the end, when we got the disease, don't really know where it came from. It came from the water that we have been changing. It came from the fry that we introduced, or even can be from some even with the feed that we are using. I one time I read a research that in the for the shrimp feed there were some some inoculums from, I think, for the from the EHP, right? So but yeah, it get complex at the end, but we had to get the start. That has to be a shrimp for a resistance for one specific pathogen. Now we enhance this immunity, and then we try to keep the environment.
Okay, thank you, Rafael, for the insight. Maybe Dr. Wahyu have another insight or opinion about this
Yeah.
question, please.
Okay. Of course, yeah, for me, the absolute priority is an integrated combination of all the three. And we know about the disease triangles, right? What it is, and this is only occurs when three conditions perfectly align, right, at the same time and place. First, susceptible host and virulent pathogen, and also a favorable environment. If we break or weaken one of these triangle, the disease cannot take hold. I think we need to for the future research, we must focus about the research priorities for integrated the three of these, not only one by one like this. We need to what it is, yeah. We need to more focus lah about the intersection of the triangle like this. Maybe maybe this is my answer.
Thank you, Dr. Wahyu,
Okay.
for the answer. One more question for Dr. Wahyu. Actually, I got one chat here. Please ask to Dr. Wahyu. Which one is more effective? I use probiotics, parabiotics, or this CGP technology?
Mana? Question, mana yang lebih efektif antara
Probiotics,
probiotik?
parabiotics, or this CGP technology?
I think depend on the target ya. CPG we know kan we directly use the what it is sequence single strand DNA mimicry in the DNA bacterial pathogen, and this like the what it is the strong booster like this, the strong booster. And if we use probiotic, this like what it is we cannot induce the immunity directly. If we use a CPG, it's like a directly inducer. This like direct inducer. You give the molecule and they stimulate directly in the short time. But if we use a probiotic, we need some time till the probiotic develop the good condition in the intestine and so on like this. And how about the paraprobiotic? Actually, paraprobiotic is the dead dead probiotic, right? And we use the wool of the bacteria, and this wool of bacteria still intake and not lysis this, and this still contain some this can act as pumps, and this also can the shrimp also can directly what it is directly mengenali adanya patogen. Ini akan dapat melatih shrimp untuk lebih waspada terhadap datangnya penyakit. Tetapi karena dia itu bentuknya wool, tidak seperti CPG yang memang sudah spesifik, jadi ya tidak sekuat atau sestrong CPG. Tetapi untuk cost production ya tentu saja lebih murah dibandingkan jika kita menggunakan CPG. Mungkin seperti itu.
Okay, thank you, Pak Wahyu, for the answer. So we still have like 4 to 5 minutes. So is there any question from the audience? If no, maybe that's all for today webinar. I would like to thank Rafael for the good information, for the sharing for us, and thank you for Dr. Wahyu. We got a lot more information about the current situation of the WSSV and how it can regulate the metabolism from the shrimp, and how this CGP ODN related technology can be a good immunostimulant for future shrimp related research or industry. So Rafael, Dr. Wahyu, thank you for today. We really appreciate the information for us. Thank you.
Terima kasih
Thank you
banyak.
so much. And yeah, I
Thank you,
heard
thank
right
you.
now we have typhoon in Taiwan.
Hmm.
I hope you are okay there.
Been raining a lot, but yeah, everything good.
Okay, maybe next time I can go back to Taiwan to visit NCKU sometimes. Thank you so much for joining us today.
Thank you.
Presentation,
Terima
Rafael.
kasih teman-teman semua. Kenapa, Pak?
Yeah, for Rafael, that's good presentation. This you have a broadened my horizons about the shrimp disease like this.
Of course. Yes.
Wahyu.
Once more time, thank you Rafael and thank you Dr. Wahyu. Bapak Ibu audiens yang saya hormati, terima kasih sudah bergabung dalam webinar kami yang ke-10. Sampai jumpa di webinar kami yang ke-11 bulan September nanti. Selamat siang dan Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Warahmatullahi wabarakatuh.
Bye.
See you, Rafael.
See you, bro. Thank you. Bye.
Okay, thank you.